webinar. It's going to be an interesting talk by Jana Sivkova about the NGS results and CNV detection in Czech patients with neuromuscular disease. Jana works as a molecular biologist at the Center of Molecular Biology and Genetics in the University Hospital in Brno. Our working group focuses on molecular genetics diagnostics of neuromuscular diseases, and she's been working there since she completed a master's degree. She's currently responsible for the implementation of the new diagnostic methods and interpretation of the detected results, which is not to say not few to do or not little to do, because it's quite an heavy task. So Jana, welcome. Thank you for accepting to be with us today and present your data. And I'm sure everybody present will be very much interested and involved, and they will be able to write the questions in Q&A, and then at the end either I read them or I will unmute the person who wants to ask you a question, and the person can voice the question. So thank you again. The floor is yours.
Thank you for the introduction. Good afternoon, everyone. I am delighted to be here today and to talk to you about our results. Yes. So in Czech Republic, there are 10 neuromuscular centers. Our Center of Molecular Biology and Genetics works within the Neuromuscular Center of University Hospital here in Brno. We, of course, cooperate with other centers, and we also cooperate with departments of medical genetics. Our center performs the molecular genetic diagnostic of neuromuscular diseases and other hereditary diseases like epilepsies, skin diseases, connective tissue diseases. As for neuromuscular diseases, we started with diagnostic of the most common types of dystrophies like myotonic dystrophy, spinal muscular atrophy, Duchenne muscular dystrophy. And then we gradually introduced the diagnostic of other genes. We used Sanger sequencing for analysis. In 2012, we introduced next generation sequencing. Nowadays, we are focusing on these groups of neuromuscular diseases, which include the most common types of dystrophies and myopathies. We also included arthrogryposis-related genes as another group. In total, we analyze 429 genes. Most of them are analyzed by next-generation sequencing. Exceptions are diseases associated with repeat expansions, like myotonic dystrophy. We used repeat prime PCR for analysis. FSHD, as disease with repeat deletion, is analyzed by Southern blot and hybridization. Next exception is spinal muscular atrophy, where MLPA method is used. And in the case of Duchenne muscular dystrophy, MLPA method is used for detection of deletion and duplication as the first step in genetic testing. Most of these genes we analyzed as the only laboratory in Czech Republic. Exception is myotonic dystrophy, spinal muscular atrophy, and Duchenne muscular dystrophy, which are also analyzed in other centers. So for NGS, next generation sequencing, we used targeted sequencing of coding regions of 429 genes, so-called panel sequencing. We use Roche technology, and for sequencing, NextSeq by Illumina. For variant calling, we use the software Sequence Pilot and CLC. To sum up our results, we examined 1,421 unrelated patients, and genetic diagnosis was confirmed in 558 patients, in about 38%. Limb girdle muscular dystrophy was the most common type of neuromuscular disease in our patients. So together with direct Sanger sequencing, we confirmed LGMD in 227 unrelated patients. 86% is the recessive form of LGMD. Estimated prevalence of LGMD in Czech Republic is 21.4 cases per 1 million. The most common form is calpainopathy. 50% of all LGMD cases is the recessive form of calpainopathy in Czech Republic. As in recent years, the dominant form has been described. We have 10 patients with late onset and mild form, and with only one mutation. In 7 cases, it's this in-frame deletion. This in-frame deletion was described by Nalamili in 16 unrelated patients, but no familial segregation was available to confirm the dominant cause. Later was described, one family was described, and familial segregation analysis and phenotypical exploration was performed, and the dominant cause of this deletion was confirmed. Our other interesting and puzzling variant in calpain is this intronic substitution. This substitution was reported several times in association with recessive form of calpainopathy, and also in recent publication is still described as pathogenic. But conflicting evidence was reported regarding the effect of this variant on splicing and RNA expression. First, CRAN reported no abnormal splicing, but later, Nestimbeni reported altered RNA splicing. We have this substitution with combination with other pathogenic variant in 14 cases, and in 10 of these 14 cases, it's in the combination with already mentioned in-frame deletion. So the question is if this substitution is needed to onset of the phenotype, or the phenotype is only due to this in-frame deletion, and it's really the dominant cause of this deletion. Now I come to the next point, which is FSHD. Its phenotype is overlapping with LGMD, so it must be considered in differential diagnosis. As you know, the FSHD is caused by contraction of D4Z4 repeats in the subtelomeric region of chromosome 4. So its diagnostic can be covered by NGS. Sudden blot and hybridization is used instead. We are the only laboratory in the Czech Republic which performs this analysis. We started with analysis in 2005, and we examined 550 unrelated patients. FSHD1 was conferred in 295 patients in about 53%. The estimated prevalence of FSHD1 is 32.9 cases per 1 million. In 49 patients with FSHD, in 41 FSHD negative patients, we performed NGS analysis and we confirmed other diagnoses in 30 of them. LGMD genes were most common. The other cause of FSHD is combination of a mutation, especially in SMCHD1 gene, and the presence of a permissive haplotype 4QA. So the diagnostic is a combination of FSHD1 testing and NGS. We confirmed FSHD2 in 4 unrelated patients. In 2 patients, it was the classic type with the number of repeats a little above the limit of 10 and with a mutation in the SMCHD1 gene. And 2 patients have a combination of FSHD1 with FSHD2. This is the example of our patient with 8 repeats and mutation in SMCHD1 gene. His family history was negative. His phenotype is mild with late onset. Now I'd like to move on the next part, copy number variation analysis from NGS data. By copy number variance, CNV, I mean the multi-exon deletion or duplications. We started with NGS with CNV analysis in 2019, and we also reanalyzed old data. We used CNV, special CNV tool within CLC software. This tool takes read mappings and target regions as input and produces amplification and deletion annotations by depth of coverage method. It's appropriate to verify these results with some other method like MLPA, genomic array, or by sequencing of breakpoints. To sum up our results, in the results, they are not included 2 genes, dystrophin and MTM1 gene, because in MTM1 gene, we detected only deletions, and as this gene is on chromosome X, the deletion is obvious. And in the case of dystrophin, MLPA method is the standard for CNV analysis and is the first step before NGS. So we detected deletion or duplication in these 10 genes. In some genes, there are described many CNVs, so we expected to detect some deletion in calpain, sarcoglycan, or laminin. But we have interesting findings in these 3 genes, and I'd like to present it in detail. Now only short note on the origin of CNV. I am not expert in on recombination and DNA repair mechanisms, so only in brief, there are two main mechanisms. The first one, non-allelic homologous recombination, needs long stretches of homology shared between breakpoints. As you can see on this picture, in the middle line, there's the sequence of deletion. Top line is the sequence before breakpoint, and bottom line sequence after breakpoint. And there's homology on both sides. In the second mechanism, there's no homology or very short homology. It's called non-homologous repair and can be divided into non-replicative mechanisms like non-homologous enjoying, and two replicative mechanisms like microhomology mediated break-induced replication. The first, I'd like to present our CNV results in the TREP-C11 gene, which is the component of TREP multi-subunit tethering complex involved in intracellular vesicle trafficking. Its phenotype is connected with limb girdle muscular dystrophy. In HGMD database, there are only 16 pathogenic variants described, and no large deletion or duplication is reported. We have 3 patients with missense mutation on one allele and with a large deletion on the second allele. These are our 3 patients. The first patient has a phenotype of limb girdle muscular dystrophy. He is 31 years old, still able walking, with proximal weakness and with age of onset at 3 years. Other 2 patients are still child, under 1 year age, under 1 year old, and their phenotype is more like congenital muscle dystrophy than limb girdle muscle dystrophy. So we tried to take a closer look at the breakpoints of these 3 deletions. There are many allele repeats within the TREP-C11 gene, so we assumed the involvement of non-allelic homologous recombination, but this wasn't present in any case. So in the first patient, we detected the deletion of 2 exons by CNV analysis, as shows this graph. There's average coverage in each exon, and you can see the decreased coverage in our patient in red color in 2 exons. We sequenced the breakpoint and found that the deletion is 2.4 kilobases in length. There's no homology within the breakpoint junction. So as you can see here, the middle line sequence of deletion, and there's only homology with the sequence before the breakpoint, top blue line, and the sequence after the breakpoint, the bottom green line. So there's no microhomology, but there is an insertion of 22 nucleotides 21 nucleotides with homology on chromosome 13. So we assume the involvement of non-homologous repair. The insertion might very likely have been copied from a dislocation by something like polymerase theta. So maybe theta-mediated enjoying is involved. In the second patient, we detected deletion of 4 exons. We sequence the breakpoint, and deletion is 7 kilobases in length, but here is microhomology present, and without any insert. So we assume the involvement of non-homologous end joint repair. And in the third patient, we detected the deletion of last 5 exons. This deletion was conferred by genomic array, as you can see in the picture. This is the deletion, and it comprises the last part of the gene and extends beyond the gene. Here the sequencing of breakpoint is still in process. So it was a TREPC11 gene. Now I will focus on nebulin gene. Nebulin is a thin filament stabilizer and regulator of muscle contraction. It has many isoforms. Its structure is highly repetitive and can bind hundreds of actin monomers. It's a giant protein with 183 exons. Within the structure, there is triplicated region, and in this region, the NGS analysis is difficult. Repetitive sequences are challenging to align accurately, which leads to imprecise basis for accurate CNV analysis on these regions, and results of CNV analysis are ambiguous. For variant calling in this region, we use algorithm for somatic mutations. So regarding the nebulin gene, we have one patient with phenotype arthrogryposis and 5 unrelated patients with phenotype of Nemalin myopathy. But interesting is that we, by CNV analysis, revealed 2 related patients, mother and daughter, with dominant myopathy. Recently, the first dominant inheritance of nebulin-caused myopathy has been reported. The identified variant was this deletion, and authors suggest the production of mutant nebulin protein with a dominant negative effect of this protein. In our family, we also detected the deletion. The deletion was detected in mother and daughter, both with weakness of distal muscles of upper and lower limbs, and with shortening of Achilles tendons. The reported case has moderate atrophy of distal muscles of upper and lower limbs. For completeness, in the third column, there is our third patient with deletion in nebulin, but with recessive inheritance. So we tried to sequence the breakpoint and found out that there's no homology, but there's insertion of 77 nucleotides with no homology in a human genome. This insertion is unusual big, and it's unlikely that these nucleotides were synthesized de novo. So most likely there must be some other source of DNA, some private DNA, or it was pieced together by multiplied jumps, and we think it's the matter of non-homologous end joining mechanism. The whole deletion is 84 kilobases in length. Now I'd like to move on the last gene with interesting CNV finding. It's titin. Titin plays several structural and functional roles in the cardiac and skeletal muscle. It's the largest human protein with 363 coding exons. Within its structure, there's triplicated region like in nebulin, and titin generates many transcripts and proteins with 6 major isoforms with different expression in cardiac and skeletal muscle. In past, the analysis of titin was difficult because its size, but nowadays with NGS, it's no problem to analyze the whole coding sequence. But the variant interpretation is challenging because of a large number of variants in titin, mainly missense variants. In GNOMAD, there are 22,000 reported missense variants in titin. However, nonsense and frameshift variants are pathogenic. Mutations in titin are related to specific types of muscle dystrophies and also with cardiomyopathies. We have one patient with limb girdle muscular dystrophy, one patient with congenital myopathy, and 2 patients with arthrogryposis. By CNV analysis, we revealed one patient with large deletion and with arthrogryposis with muscle dystrophy. Deletions and duplications, large deletions and duplications are rare in titin. In HGMD database, there are only 4 large deletions reported, 2 with dominant and 2 with recessive phenotype. In our patient, we detected deletion, only deletion, no other mutation, but the dominant cause can be confirmed because we don't have both parents for segregation analysis. The main clinical symptom of this patient is arthrogryposis. So by CNV, we detected this large deletion. By sequencing of breakpoint, we found that this deletion is 44 kilobase, and there is a microhomology in the breakpoint. So we think it's the matter of non-homologous end joining. So let's move to the next part. As I mentioned at the beginning, we include arthrogryposis related genes in our panel, as these congenital contractures are caused by decreased fetal movement during development. Muscular dystrophies and myopathies are the common cause of arthrogryposis. So we tried to offer a comprehensive diagnostic of arthrogryposis. So we include genes related with distal arthrogryposis and connective tissue diseases. My first example is the example of association of all gene with a new phenotype. SCN4A gene is usually connected with periodic paralysis and myotonica congenita with dominant inheritance, and with congenital myasthenic syndrome with recessive inheritance. We had two cases of fetus with severe contractures and polyhydramnios. In the first case, the baby was born at week 32 and died in 10 days. In the second case, there were two pregnancies from one mother, both with the same ultrasound findings, and both terminated. In both cases, we found two mutations in SCN4A gene, so the recessive inheritance. In literature, we found several cases also connected with this severe congenital myopathy. So our result extends the phenotypic spectrum of diseases connected with SCN4A gene. My second example is a boy with arthrogryposis. His clinical picture is dominated by dystrophic habitus with muscle weakness, contractures, scoliosis. Muscle enzymes were always normal. His initial diagnosis was congenital myopathy, but later re-evaluated for arthrogryposis. We performed the first genetic testing at his 3 years, but with no result. Later, as we included arthrogryposis genes in our panel and introduced CNV analysis, we repeated the genetic testing, and we detected homozygous duplication in PLOD1 gene. So this is the duplication detected by CNV analysis. We confirmed this duplication by MLPA method. So the final diagnosis of this patient is Ehlers-Danlos syndrome, whose associated genes are not normally included in neuromuscular genes. And in the last part of my presentation, I'd like to show that inheritance pattern doesn't have to be as it seems to be. Regarding the laminin, we have 20 unrelated patients, mainly with phenotype of congenital muscular dystrophy. But we have interesting case, two families with recessive diseases in 2 generations. So The first, it looked like dominant inheritance. By NGS analysis, we detected 2 mutations in laminin in the child, but the carrier status of mutation in laminin in father didn't explain the father's phenotype. So we sequenced the whole gene and found other mutation. Very similar is the situation in the next family. I think it's important to be aware of these unusual cases. For example, when whole exome sequencing is performed in trios, and the data are then filtered according to the expected inheritance. So the expected inheritance in this family would be dominant, and it would look like only that the child and father will be only carriers of mutation in laminin. So to sum it up, for routine diagnostics, we still prefer panel sequencing to whole exome sequencing. The main advantage for us is higher coverage, so better input for CNV analysis. As we designed this panel, we have better knowledge of included genes. Of course, the amount of data is not so large. The big disadvantage is that only the selected genes are analyzed. CNV analysis, as our results showed, CNV analysis should be included as integral part of NGS data evaluation. In a set of our patients, CNV analysis allowed identification of large exon deletions in TRPC11 gene as the cause of muscular dystrophy. No deletions have been published yet in this gene. Next, identification of large exon deletion in nebulin gene as the cause of dominant myopathy. And together, we confirmed, thanks CNV analysis, genetic diagnosis in 1.1% of unrelated patients, which is small percentage, but significant for the patient and his family, as the definitive diagnosis is confirmed, and thus enable genetic counseling in the family. And that brings me to the end of my presentation. I'd like to thank all colleagues from our center for their kind cooperation, especially our leader of our neuromuscular group, Lenka Fajtusová, my colleagues Tereza Kramářová, Johana Kopčilová, Zuzana Hrubá, for their help in laboratory and with data evaluation, Kamila Réblová for bioinformatics support, and Jan Svatoň for sequencing service. And I'd like to thanks collaborating neurologists and medical genetics, especially Jana Haberlová, Radim Mazanec, Stanislav Voháňka, Andrea Grzegorzowa, Petr Ridzoň, and many others. And thank you for listening.
Thank you, Jana. It was a very interesting presentation. I'm glad that the questions are now appearing on the box. So Joseph Canes, you can talk if you want, just unmute yourself and put your question, otherwise I can read it. I don't know if he's in difficulties for that, so I'm going to read it. First of all, he congratulates you for the nice presentation, and then he asks, what TNPO3 did you find in the LGMD2 case? Do you have clinical information for this individual, and did you test the variant in the parents?
I think we have one patient with TNPO3 mutation, but I'm not sure if we have clinical information. And if we tested the parents.
So Marco Savarese, you can unmute yourself and you can speak up, please.
Hello, can you hear me?
Yes, perfectly.
Yes, okay, thanks a lot. Very interesting talk, very interesting findings. I have a couple of questions. The first one is, if I understood correctly, you are only evaluating multi-exonic copy number variants. Do you have any plan for single exon copy number variants and how to detect them?
Yes, single exons, we only look at single exons copy number variants in case where we have one other mutation in this gene, and we suppose this gene. Because if we look in general, we have many false positives results. So only in individual cases.
Okay, thanks. And the second question is, do you also have access to muscle rendered by RNA, and are you planning to eventually evaluate the effect of the identified copy number variants on the transcripts?
Yes, we plan RNA sequencing in future, but not yet.
Thank you. And the third is not a question, but we have a very similar finding in titration, but I'm going to eventually mail you and discuss it. I have to admit, I'm not sure that I don't have any email. If you can eventually, because we have a very similar finding in titration.
Yes, okay.
So we can discuss that.
Try.
Yeah, you can write your email on the chat if you don't want to send your email to everybody. You can send it to Marco Zavarese.
Okay, yes, he was in the first. Sorry, yes, I missed it.
Can see the master slide?
Yes, thank you so much. Yes.
Okay.
Okay, excellent. So let's see if there are new questions. No. Okay. Then I'll have a question. Is it me, or did I understand that your prevalence of limb-girdle muscular dystrophy is the double of the average from the OrphaNet database?
Sorry, I didn't understand.
Did I understand correctly that the prevalence of limb-girdle muscular dystrophy in your data is the double of the average that is in the mentioned by OrphaNet?
Yes.
You said that you had 2.4 per million, wasn't it?
Yes,
And
we have, yes, and in OrphaNet it's 10 to 9 cases.
21 for, yes, yes. So what do you think is causing this number to pop up? Is there is an epidemic of limb-girdle muscular dystrophy in Czech Republic, or it's another explanation?
I'm not sure. I think I don't know.
Do you have referrals from many regions, as you said in the beginning? Is that a bias from the referrals that you receive, perhaps?
Maybe.
Okay, good. Anyone has more questions? Right. So the TRAPS-11 adult patients, did they have an history of congenital or infantile or juvenile symptoms of the disease or not?
In TRAPS-11 gene, we have only these 3 patients. Yeah.
One is an adult and 2 are babies, so very young guys.
Yeah, they are too young.
And they have an hypotonia and they have a congenital presentation. What I was asking is the adult, does the adult has any symptoms of early onset or not?
I think the onset of the adult was at 3 years. So it was later than in these 2 cases.
Mm-hmm.
And
and did you look at the databases, the usual ones, to see if anyone had described any further cases, or that was excluded?
We only looked if there was described any deletion or duplication connected case. So, but of course there are described cases with other types of mutations.
Okay, so in terms of what you've described and you showed today is an impressive amount of work. And what I wanted to ask you is, what difference do you think that diagnosis capacity has done in terms of treatment for those patients? Do you think it changed the way they were cared for afterwards? Did they start having, for instance, cardiological counseling for the ones with type 2, for instance, or how do you think that the treatment of the patients has been modified, in case you have any feedback regarding that, of course?
So I can't say it exactly, but I hope that the treatment will be different. But I think that I think that the feedback of this is mainly for the family and the genetic consulting, because there's no special treatment in these cases.
Mm-hmm. Okay. Since we don't have any other questions, I think that I'm going to thank you for your presentation. Thank everybody for attending this meeting. As you know, these webinars are recorded, and you will find the recording in the webinar section of our website, and also in our YouTube channel, Euro-NMD. So either you or someone else wants to see what was presented today, you just refer them to one of those sources, please. And thank you very much, Jana. Thank you very much, and hope to meet you very soon. Okay?
Thank you.
Bye now.
Bye-bye.